Hello, my name is Charlie Pophal. I'm part of the Science Internship Program at the Cleveland Clinic. This summer, I worked at molecular cardiology. I'm going to be a junior at Ignatius. My project is novel regulation of cardiac hypertrophy modulating protein glycogen synthase kinase 3. The overview. Congestive heart failure occurs when the heart cannot pump enough blood and oxygen to support other organs. Affects estimated 5.8 million people in the U.S. About 20% will die within 1 year from the diagnosis. One type of this heart failure is caused when cells stay in this stress state, which causes the heart to work more and eventually fail. And this is the type that I looked at. Molecular interactions that are caused by stress. When a person is under stress, there's enhanced energy utilization that comes along with increased cardiac output. The increased output chemically is associated with direction of multiple cellular signaling pathway, accounting for enlarged cardiac cells and cardiac hypertrophy. And as you can see, the bottom left picture is of beta-adrenergic receptors, which are located on the cell membrane. And the right is the pictures of receptors which signal to the heart during the fight or flight stage for more blood. Direction of cellular pathways is associated with increases in kinases, which are types of enzymes. Phosphatases, which are another type of enzymes, normalize the increase made by kinases. Therefore, a stressed cell has a higher phosphorylation state, which is a more stressed state, due to the elevated kinases. Now here's a little graph of what I made. As you can see on the left, the cell is normal or dephosphorylated. The kinases bring it over to the phosphorylated state or the stress state, and the phosphatases bring it back to the normal state. And this is an example of normal phosphorylation. An enzyme called PI3K disrupts normal dephosphorylation between PP2A and PPMT. PP2A is the enzyme that is a part of dephosphorylation, and PPMT activates PP2A. When PPMT and PP2A don't interact, there isn't dephosphorylation. PI3K keeps the cell in that aggravated state by inhibiting the enzymes PP2A, forcing the cell to remain in the agitated state. However, PI3K positively regulates protein function. And here's a graph showing PI3K inhibiting PP2A. Now, as you can see, this is similar to the one before with a few changes. When the cell is normal or dephosphorylated, the kinases bring it to the phosphorylated state, which is the normal way it usually goes. The problem comes when the phosphatase tries to bring it back, but the PI3K blocks the phosphatase PP2A, keeping the cell in the stress state, causing heart problems. The chemical warfarin is a PI3K inhibitor. At its introduction in the early 90s, it was expected to help cancer patients by inhibiting PI3K. Its inhibition had unintended consequences as PI3K biology was not completely understood. Goal for the future is to find a PI3K inhibitor that does not deter PI3K protein functions. Now, my hypothesis for the project was that PI3K interrupts dephosphorylation by disturbing PPMT, which directly disrupts the phosphatase PP2A. My methodology. I started off by using a process called Western blotting. What I did was I took human embryonic kidney cells and I grew them in 4 plates using something called media, which helps the cells grow. I then added warfarin to 2 plates, and then I added LY29004 in other parts of the experiment. Warfarin and LY29004 are both extremely similar and both have the same effects. They're both PI3K inhibitors, and so in the experiments we use them to test PI3K inhibition and its effects on PP2A and PPMT. Next, I added DMSO to 2 of the plates. DMSO is a control and does not disrupt PI3K. Following the above treatment, I added isoprotanol to one Wortmanin/LY29004 plate and one DMSO plate. The other Wortmanin plate and the other DMSO plate I kept as a control. Finally, I estimated how much protein is in each amount of cells. I then added PP2A to it. I made a gel and added the cells of different wells in the gel. I ran the gel through a Western blot to transfer the cells using electricity from the gel onto a membrane. I blotted for PP2A, PPMT, and PI3K. I blotted using films in the darkroom, which provided one view of the data. I used the same procedure and I repeated it on small glass slides, which I did for my next experiment. My next experiment was confocal microscopy. In this, I used 6 slides I talked about before that were placed in different inhibitors, 2 Wortmanin, 2 DMSO, and 2 PI3K knockdown cells, although the PI3K knockdown cells did not work. And I kept this in for several days. I then added a green fluorescent dye, which made it easier to see the different cells. The slides were viewed with a confocal lens or confocal microscope. The confocal lens is so powerful that it can take pictures of the individual cells so you could see phosphorylation in the different cells. Here are my results. This is a magnified image from the PP2A methyltransferase blot, which shows an intense PP2A band in the Wortmanin wells compared to the DMSO wells. This is because there is more interaction between PP2A and PPMT since PI3K is inhibited, and it proves the hypothesis. So as you can see, the DMSO control has a less intense line than the Wortmanin control, and the DMSO isoprotanol has a less intense line than the Wortmanin isoprotanol. This blot is a PI3K with a DMSO control and Wortmanin control not being involved because of error. As you can see, the DMSO has a darker band of PI3K, while the Wortmanin has a lighter band, showing that PI3K has been inhibited on Wortmanin. This is a magnified image from the PPMT blot, which shows an intense PPMT band in the LY29004 inhibitor, which is an inhibitor very similar to Wortmanin. And these wells compared to the DMSO wells. This is because there's a robust interaction between PP2A and PPMT since PI3K is inhibited. This view also directly correlates with my hypothesis. This is a confocal image of phosphorylation. As you can see, the inhibitive cells have much less phosphorylation than the control. This proves that PI3K directly interrupts dephosphorylation by disrupting PI3K. My conclusion: when inhibitors of PI3K, Wortmanin, and LY294002 are used, PP2A and PPMT interaction is not disrupted. PI3K directly prevents PP2A and PPMT from interacting, which disrupts the dephosphorylation process. Recommendations: although our hypothesis was proven, larger studies of this material are recommended to improve statistical significance. And we need to carefully handle the cells so they are not contaminated, which was a constant problem in my experiments. Finally, by continuing to study PI3K and its relationship with PAP2A, PPMT, a greater understanding of treating congestive heart failure could be obtained, helping the many people with the disease. Acknowledgements. For my project, I would like to thank Rosalind Strickland, Nedra Starling, the Office of Civic Education Initiatives, my mentor, Satya Malanga Prasad, my co-mentor, Mayor Dumaine Mohan, and my other co-mentor, Elizabeth Martelli. And thank you very much.